PMSF, or phenylmethylsulfonyl fluoride, is a chemical compound that plays a significant role in biochemical and molecular biology research. It is a serine protease inhibitor, targeting a specific class of enzymes, called serine proteases, that are responsible for breaking peptide bonds in proteins. The chemical structure of PMSF consists of a phenyl group attached to a methylsulfonyl group, with a fluorine atom replacing one of the hydrogen atoms on the phenyl ring. This unique structure gives PMSF its potent inhibitory properties. PMSF works by irreversibly binding to the active site of serine proteases, forming a covalent bond with the serine residue present. This binding causes a conformational change in the enzyme, rendering it inactive. The inhibition of serine proteases is vital in various biological processes, including digestion, blood clotting, and immune response. By using PMSF, researchers can investigate the functions of serine proteases and their involvement in specific pathways or diseases. To use PMSF effectively, it is typically prepared as a stock solution in an organic solvent, such as ethanol or isopropanol. However, it is important to note that PMSF is highly reactive and sensitive to moisture. Therefore, it should be stored in a tightly sealed container and kept away from water sources to maintain its stability. In laboratory experiments, PMSF is added to protein samples or cell lysates to inhibit serine proteases. The optimal concentration of PMSF depends on the specific protease being targeted and the experimental conditions. Typically, concentrations ranging from 0.1 to 1 mg are used. However, it is crucial to handle PMSF with care. It is considered hazardous and can be toxic if ingested, inhaled, or absorbed through the skin. Therefore, proper personal protective equipment, PPE, including gloves, lab coat, and safety goggles, should be worn when working with PMSF. It should also be used in a well-ventilated area or under a fume hood to minimize exposure. While PMSF is a widely used serine protease inhibitor, it does have some limitations. It is not effective against all serine proteases and may have varying degrees of inhibition depending on the specific enzyme. Additionally, at higher concentrations, PMSF can also inhibit other enzymes, such as cysteine proteases. Researchers may consider using alternative inhibitors, such as AEBSF or TLCK, depending on their experimental needs. In conclusion, PMSF is a potent irreversible inhibitor of serine proteases, playing a crucial role in biochemical and molecular biology research. Its ability to covalently modify the active site of these enzymes provides valuable insights into their functions and involvement in various biological processes. However, it is important to handle PMSF with caution and consider alternative inhibitors when necessary.